Yeah, I wasn't sure if you had left yet.
Is there any sound
Oh,
in the
okay.
presentation?
If you can
Okay.
bring a mouse.
Yep, that's all you need to
just in
do.
case anything goes wrong, have
The
to
light's
track
already
off it.
good. Let me just give it a second and I'll make sure
If
that
it's
it's
possible.
working.
If it's not, don't worry about it.
So right before she goes, just hit start.
Actually, I've already
Oh, you
started
already
it, so
done
you
it?
should be
Oh,
fine.
okay.
Yep.
Okay.
If
Sounds
you
good.
want.
Yep.
I can tap this instead.
Okay.
Besides that, you should be all good though.
Hello. Okay, I don't want to get confused between the points.
Can you make a plug for Delaney's talk at 11:05?
Yeah. Like, you could be like, whoa.
What's up?
One baby eats, that's no lie. And this is my sister Anne. No, I'm not gonna do that.
No, it's just, there's, I honestly think this could be fun. This could be the end of it. No, we've talked about this. You especially, is there a huge thing for things about people? No, genetics are real. If you're intelligent enough to take over the body, underlying the body.
Who'd you meet with this morning?
I think I was long enough that my body rejected alcohol. I think you can tell the difference between like 40 and 10. It's all horrible. It's all just screw this. No, I don't Yeah, I was so excited. He's got this like superpower.
And the podium, like I literally, they had like a little stool for us to stand on. So you could see over the podium. They were like, that's so short. The thing is, when you done this on the podium, like I was like this. No, if I didn't, if I wasn't standing on the stool.
I know.
So I know if you put it so let's see.
This one is like,
Yeah, it's really good.
Now, if your gene is in the spinal blood, you put the locks inside of the let's say your gene is in the movement can you put it in the spinal or you have to find where the or is it just like magic?
You're like back here, right? And so you can't see that.
oh, yeah. I realized that.
to write everything down. I cannot remember stuff.
Oh. Yeah. Like, let me remember your life story. Where's your career at?
I told myself I wasn't going to be allowed to today. I'm like, yeah, I literally was like,
Okay, guys, it's 12 o'clock. We're going to go ahead and get started so we have enough time to listen to Anne. Matt, you particularly need to pay attention to this. So before we get started and I introduce Anne, I actually would like to make a shameless plug for the Pioneer Seminar Series that's also happening in Roanoke this week. Our speaker out there is Phil Hayden. He's there on Friday. His talk will be live streamed from 11 to 12 for those who are interested. Phil studies, he's at Tufts, he studies glia and astrocytes, and I'd say what he's been working on recently is sleep. I don't know if that's what he's going to talk about, but he gives a great talk. Okay, so today, that's not what I'm here for. I'm here to introduce Anne Gregus. Many of you know Anne already. She's been at Virginia Tech for the last 2 years, and I have the pleasure of introducing her today. So I've got my notes. I'm going to tell you a little bit about Anne. So Anne got her BS in biology in Pennsylvania in Villanova. In 2008, she got her PhD in pharmacology in the Weill Cornell Graduate School. She investigated mechanisms of opioid tolerance and drug reward, and this has kind of been her path. She's followed this throughout her PhD, her postdoctoral fellowship, her time in industry. I'll spend a minute talking about that, and now the work that she's doing now. She was a postdoctoral fellow from 2008 to 2013 at UCSD. And there she employed metabolomics approaches to identify spinal microglial 1215 lipooxygenase, and that's a lot, yes, as novel endogenous pain signals. So following her postdoctoral fellowship at UCSD, she went to LPATH, and she worked in industry. And she did a lot of things there. So she was busy. But basically she worked on this bench to bedside, studying, bringing novel targets or therapeutics. I don't know why I'm so nervous right now. This is crazy. You're making me nervous, Anne.
Be nervous.
You know, I would say she's one of the few faculty members that we've interviewed who has industry experience, which I think is a real plus for the type of research that she's doing, the stuff that she's going to talk to you about today. I think it's really great for our graduate students to see that you can have alternative career paths. And as somebody who runs a graduate program, she's already come and talked to some of our students about that path, which I think is, really, really useful. So since coming to Virginia Tech, she got here in 2016. She came as a staff scientist, and her work has carried on studying pain, looking at non-opioid drug targets for neuropathic pain. She uses a wide array of techniques, including click chemistry, mass spectrometry, metabolomics, and GPCR omics. Since she has been here, she has applied for and obtained R01 funding. She also has funding, an equivalent to R21 funding through the state of Virginia. In addition to her science, Anne has been involved in teaching at both the undergraduate and graduate students. She has trained undergraduate students in her lab. These students have won awards, so she's been a great ambassador, I guess would say, for our program. During our Skype interview, we Skype interviewed Anne like we did our other candidates, and she talked about new courses that she could develop. And so with that, I'll just say that I think Anne is a great steward. She's a great colleague. We all enjoy having Anne here. And so with that, I'll introduce her and stop talking like this with my voice being all shaky. Like this is the first time I've spoken in front of an audience. The title of her talk is Preclinical Target Refinement of Novel Non-Opioid Therapeutics for Chronic Pain States. Thanks.
Thank you, MC Baby Ace. Dropping some knowledge on all of us. I'm so excited to be able to talk to you about what I've been working on, to see so many familiar faces in the audience. Thank you so much for coming out. And I'd like to thank the School of Neuroscience for inviting me here to talk to you today. So I've been studying pain for the better part of about 20 years. And, in the context of academics and in industry, and I've been focused on this idea of preclinical target refinement. And the reason for that is because so many drug candidates fail in phase 2, which is generally when they're tested for efficacy. So this is about maybe 2% of all molecules actually make it past this phase. And the question is, what is the reason for that, right? So this could be dependent on, how we set the clinical trial parameters, the endpoints, the readouts that we look at for those trials. And it could also have something to do with the targets that we select at the preclinical level. Is there some deficiency there? And so my hope in coming back to the academic setting was to, somewhat improve the rigor in this early drug discovery stage area. And I'm going to talk to you specifically about discovery of novel non-opioid targets, because as you know, we have an opioid crisis, and there's definitely a need for alternative therapeutics that are effective for chronic pain states. Okay, so as I'm sure you probably know, chronic pain or maladaptive pain is a major health issue. It affects approximately 1/3 of Americans. So look to your left, look to your right. If it's not one of them, guess what you... It's fine, just stand next to me and your odds will improve. So this is a huge economic burden to society. It costs about $600 billion a year in lost wages and in medical costs. This is a burden that is greater than that of heart disease, cancer, and diabetes combined. So as you can see, this is a pretty significant problem. 75% of patients, particularly those with neuropathic pain, report that their pain is poorly managed. This means that currently available therapeutics that we have are simply not effective. As you might know, the two main ones are opioids and nonsteroidal anti-inflammatory drugs. The majority of the other drugs that are prescribed are actually off-label, so they're used for epilepsy, they are used for depression, etc. And so this is a very underserved area. It is also often comorbid with other disorders, including mood disorders, sleep disturbances, which you could imagine would then further exacerbate the pain state. And as I mentioned before, we have the opioid abuse crisis that has been declared a public health emergency, and so we need clear alternatives. Some people have decided to study biased opioid ligands, where you have a drug that will activate the analgesic activity, perhaps without causing some of the side effects or the addictive-like properties. I've chosen to focus on novel non-opioid mediated targets for pain. Okay, so acute or nociceptive pain actually serves a protective or physiological function to prevent further tissue damage. So this is sort of a basic cartoon of what happens when you have noxious mechanical, heat, and chemical stimuli that are transmitted along axons. These are either of the A-delta thinly myelinated variety, or the C fibers, which are unmyelinated. As you can see, they conduct impulses at different rates. These are transmitted through ganglia that are in the peripheral nervous system. These can be dorsal root, trigeminal, or nodose ganglia. Dorsal root is at the level of the lumbar cord, which we study in mouse models. These are transmitted into the spinal cord dorsal horn, where they are modified by interneurons, by incoming signals from larger A-beta or non-nociceptive fibers, and transmitted to higher order centers in the brain. These signals are also shaped by incoming descending inputs from the brain as well. Oops, I'm sorry, I'm going in the wrong direction. Okay, so an example of an acute nociceptive pain stimulus, I'm sure you've all done this. You've stubbed your toe on a table, and basically this, the pain response is supposed to tell you to stop doing that so that you don't further injure your toe or, maybe break your leg. You might be familiar with the feeling after that. So you have this, sort of sharp stabbing pain. Those are your A delta fibers. Those conduct signals more quickly. And then you have that followed by a sort of latent burning, throbbing sort of pain. These are your C fibers. Those conduct impulses more slowly. And you might be saying to yourself, okay, well, why don't we just get rid of pain entirely? So the consequence of that is that you can develop CEPA, which is congenital insensitivity to pain with anhidrosis. So basically, you cannot sense pain signals normally, and this results in significant tissue damage and reduced lifespan. So you could imagine that that's not really the solution, right? We need to be able to, block maladaptive pain. Okay. So the major question in the field is, how does acute pain become chronic? And that's not to say that if you stub your toe that you're going to develop chronic pain. It's obviously more complicated than that. So generally you have some sort of insult. You have an injury, malignancy due to cancer, some sort of infection or metabolic disorder that changes the wiring, you would say very simply, of both the peripheral and the central nervous system. This is essentially turning up the gain on how these signals are interpreted. And, the mechanisms of this are fairly complex. They involve, activation of ion channels like NMDA receptors, phosphorylation of kinases, activation of CREB, which is a transcription factor. And basically, the sum of this is that these signals are amplified. This is a process that's known as central sensitization. And you can think of it as sort of forming a pain memory. And in fact, a lot of those processes are very similar to long-term memory formation in the hippocampus. Okay, so an example of these different pain states occurring together is rheumatoid arthritis. So I should tell you, there are different types of pain. There's the nociceptive pain that I described before, inflammatory pain, where there's obviously some sort of inflammation, neuropathic pain, where there's some kind of lesion to the nervous system, and there's also a sort of what is called idiopathic pain state, where we don't really know what the cause is. Those are things like fibromyalgia. So rheumatoid arthritis is a good example of pain states occurring simultaneously. So what happens here basically is that, you have an influx of immune cells into the joint after there's a break of immune tolerance. And you have essentially like a degradation of the joint. You have thickening of the synovial membrane, increased expression of cytokines and things in the synovial fluid, and a degradation of the cartilage and the joint itself, and this results in chronic pain. So it turns out this is an autoimmune disease that has a 0.5 to 1% incidence worldwide, and women are 3 times as likely to have it, so lucky us. So as you might imagine, there is an importance when you're doing these animal models to include females in your studies. Historically, we've studied males mostly, and the field is definitely moving towards that, which is good because, as you might imagine, those therapies might be unique to women versus men. Okay, so rheumatoid arthritis pain is undermanaged. Basically, what happens generally in the humans and in the animal models is that you have an inflammation that is sensitive to your non-steroidal anti-inflammatory drugs, or NSAIDs. You've probably taken these. These include ibuprofen or aspirin, and this inflammation resolves. And basically what persists beyond that is a neuropathic pain state that occurs in the absence of a peripheral inflammation. And we call this transition phase the chronification of the pain state. So it turns out that the inflammation itself is reasonably well controlled at this point. So we do have NSAIDs, as I mentioned before. We have these disease-modifying anti-rheumatic drugs, or DMARDs. These generally include your biologics. An example of this is etanercept, which targets tumor necrosis factor alpha receptors. And this also includes the newer Janus kinase inhibitors that were just recently approved. These also might include things like steroids. Maybe you've taken prednisone for severe inflammation. You might also know that taking these is incredibly unpleasant. You will be moody and have weight gain all at the same time. And so, that's reasonably well controlled, but it turns out that the neuropathic pain state is really not. We're limited in terms of effective treatments here. essentially you have an anticonvulsant gabapentin, which I would argue, and other people would as well, that it has pretty limited efficacy. It works generally in maybe about 30% of patients, and it's very short acting, right? So you've got to take this 3 or 4 times a day, and many times people forget because who's going to remember to take a pill 4 times a day, right? So it can be difficult. And so there's a clear need for alternative treatments here. So just to give you an example of the things that we measure in both animal models and in humans are different pain states, and these are known as allodynia and hyperalgesia. So a good example of this is a sunburn. So we have Batman. He's at his summer home. Really? Nobody's laughing about that? Come on, man. And so, he's used to being in his bat suit. He doesn't see the sun very often, and so he gets a sunburn. And so what happens is, basically, a stimulus that is not normally perceived as painful is now perceived as painful, and this is called allodynia. So when he puts his bat suit back on, he feels very, very uncomfortable. And, this is something that is seen particularly with cancer patients. They feel like, okay, even just putting my shirt on is horribly painful, right? And you also have hyperalgesia. This is in which a stimulus that is normally considered to be noxious is now perceived to be as more painful. So if he gets, punched in the face with a sunburn, then he's in even more pain. Thank you, Matt, for that analogy. Okay, so we can model this in animals. One example of this is called the KBXN serum transfer model of arthritis. What happens is you breed a certain mouse lines together, and you obtain a KRN arthritic mouse, and you can obtain the serum for that and actually inject it into another mouse. So this could just be in a C57 Black 6 mouse. You can imagine you could then take advantage of genetic tools in those mouse lines. And so what happens is, of course, you have peripheral inflammation. You can see right here is an ankylosed pulse. So the digits are actually pointing to an angle here. This is characterized by an inflammation that peaks at around about a week or so and then resolves within about 3 weeks. That is also accompanied by allodynia. That's what I described before, is the bat suit with sunburn. We measure this by using the von Frey test, where you present a series of filaments that are of increasing graded forces, and you measure the threshold at which they withdraw the paw from that stimulus. So you can imagine, in the absence of injury or with a control serum that's non-arthritic, there is no change in baseline thresholds, but after administration of the serum, they develop a severe allodynia. So that's shown by a reduction in the threshold. And if you'll notice, that persists beyond the resolution of inflammation. So this is clearly a problem that needs to be dealt with, and it's something that we see in humans as well. So we wanted to explore the mechanisms behind this. And I'll save you a lot of trouble because this is published, but essentially we were interested in the innate immune system. So TLR4, or toll-like receptor 4, is something that's present in immune cells. It's part of the innate immune system. Georgia could certainly tell you, she could do a whole seminar on this, I'm sure. And downstream of that, you have activation of cyclooxygenases, which I told you before are blocked by NSAIDs, or non-steroidal anti-inflammatory drugs, and TNF-alpha, which this process is blocked by etanercept, which I mentioned before is used in patients as well. And so we wanted to investigate these mechanisms in both the process of the inflammation and in the chronic pain state. And so we used a model of a knockout mouse for TLR4, a constitutive knockout, and we also used antagonists. And what we found was that if TLR4 is knocked out, it does not affect the inflammation at all, but it does completely reverse the allodynia. And you'll notice that it happens, around this period of about 3 weeks when the inflammation is resolving. So it seems to play a role in this chronification phase. We address that further by using antagonists that were delivered spinally for TLR4 and found that, I'm not showing you this here because it's all published, but if you inhibit TLR4 at this, prior to this chronification phase, you reverse allodynia. But if you do it after that, if you do it later, about 3 weeks or so, it has no effect on the allodynia. So TLR4 is clearly playing an important role, at this chronification, at this acute to chronic pain transition point. So we also asked that question by using NSAIDs and etanercept. And again, I'm not showing it here, but it's published. What we found was, yes, those affect the inflammation and the early phase of allodynia, but there was no effect on the chronic pain state. So those actually failed in the animal model as well as in the humans. So we were pretty confident that this is a good model to not only mimic rheumatoid arthritis in the human, but also for us to investigate the pharmacology of that and to look at possible targets that we might intervene with drugs. So we were also able to model this transition phase in the rat as well, and that was important for us to be able to show, sort of a translatability between species. That's something that's important in drug development as well. You want to show that the mechanism is something that you can recapitulate in multiple animal models. And what we found was, again, if we injected spinally, intrathecal administration of lipopolysaccharide, or KLA, which is KDO2 lipid A, that's the active form of LPS, we produced a significant allodynia. Shown here is the area under the curve, so maybe you can see a little bit more easily into the, maybe amount of pain-like behavior that's there. You can see with vehicle, this is a low number. This is increased significantly with KLA treatment, and this is not reversed by ketorolac, which is another NSAID. So the important point about this is that this pain state is not driven by cyclooxygenase metabolites, right? So this is potentially not something that's going to be influenced or affected by anti-inflammatory drugs. However, it was sensitive to inhibitors of glial function, and there's some argument about how specific or selective these are. One, minocycline, people have shown to be more interactive with microglia. Pentoxifylline is sort of a general glial inhibitor, but both of those reduce the allodynia that was caused by intrathecal administration of KDO2 lipid A. So this pain-like behavior is dependent on glial activity, but not on cyclooxygenase metabolites. So the question is basically what's going on here, right? And we asked the question, what are the downstream signals from TLR4 activation in the spinal cord that are happening in this pain state? What is driving this pain state? So we decided to study lipids. Those are things that are activated significantly following TLR4 activation, and you might ask why, because it's a little bit complicated, but it turns out that cyclooxygenase inhibitors block lipid metabolites coming from a substrate known as arachidonic acid. So this is an omega-6 fatty acid. You might be familiar with the omega-3 fatty acids, fish oil. They're also be very good for you. All that means is that the first double bond is 6 positions from the omega end. I know, it's blowing your mind right now. So what's interesting is that for decades, people thought that not only, that arachidonic acid could come from your diet, but that it could be released from the plasma membrane as well by an enzyme called cytosolic phospholipase A2. And it was thought for decades that the majority of arachidonic acid was supplied by this enzyme. And some recent very elegant work coming out of the labs of Ben Cravatt at Scripps and Aaron Lichtman at VCU, they have shown that a significant amount of arachidonic acid is actually generated from 2-arachidonyl glycerol, which is simply arachidonic acid with a glycerol in it. I'm not showing the structures because I don't want anybody to throw things at me. Through monoacylglycerol lipase and anandamide, or arachidonyl ethanolamine. So this is just arachidonic acid with an ethanolamine. And that's degraded by an enzyme known as FA, or fatty acid amide hydrolase. So arachidonic acid can then be processed by cyclooxygenase enzymes, as I mentioned before, to form prostaglandins, among other things. These are things that are involved in fever. Maybe you've taken an aspirin to block a fever. That's reducing the effect of prostaglandins. And as I said before, they're targeted by NSAIDs. And some of the newer generation COX-2 inhibitors, you may have heard of Celebrex. So another pathway that arachidonic acid can be processed by is 5-lipoxygenase. So these make your leukotrienes. So anybody who has allergies, maybe you've taken Singulair, these things will block leukotriene receptors or the enzyme itself. So we also have cannabinoid receptors that can be activated by our endocannabinoids that I just described here. So one of the drugs on the market, Marinol, I believe that's for nausea with chemotherapy. And then of course there's, now a drive towards legalizing medical marijuana. There are different opinions in terms of the efficacy and the side effects. Being a lipid, it's very hard to dose this, so we'll see what happens with that. But there are drugs that are selectively targeting MAG lipase and Fa that are now in phase 2. There are also cytochrome P450s that process arachidonic acid. These tend to be your liver microsomal enzymes, so it's not common to target them because they're typically involved in drug metabolism, and it's just a really bad idea. There were a couple of drugs that were actually pulled from the market for severe interactions that way. So there is a company called Ecosis that's focused on blocking the breakdown of the EETs that are formed from cytochrome P450s because they're thought to be, anti-inflammatory, pro-resolving type mediators. And so that leaves us with one more pathway. It's also lipoxygenases, but these are the 12 and the 15 lipoxygenases, and this is less well studied. We have shown that they influence inflammatory pain, but we were interested to see if they would also be involved in this, neuropathic pain states. And for anyone who's interested, the 5 and the 12-15, that just means the position, the stereospecific position on which oxygen is inserted into the arachidonic acid molecule, and that's all I'm going to say about that. Okay, so we needed to be able to identify these TLR4 dependent bioactive signals, and we did this with mass spectrometry. So we did this in 2 different models. So when we intrathecally administered LPS, TLR4 agonist, and we harvested the spinal tissue at different time points that were correlating with the pain-like behaviors. And we also harvested and cultured microglia and astrocytes because, as I told you before, there was some sensitivity to block of the pain state by pentoxaphylline and minocycline. And then, so we cultured those cells and exposed them to lipopolysaccharide and then harvested the media. So we ran all these in a targeted lipidomic analysis, which included almost 200 lipid species of these pathways that I described, the cyclooxygenase, the lipoxygenase, and the cytochrome P450. You can see some sample traces here. And so here is a characteristic heat map. You may have seen this for genomic type studies. So basically what we have here are your 5-lipoxygenase and 12 and 15 lipoxygenase metabolites, the cytochrome P450s and non-enzymatic metabolites. I did not include the cyclooxygenase metabolites even though we measured them. This was done in spinal cord tissue and also in microglia and astrocytes at different times after treatment with lipopolysaccharide. And we used Benjamini-Hochberg false discovery rate to sort of basically focus the analysis on targets that were only significantly upregulated and that were not part of the cyclooxygenase pathway, that were altered in the spinal cord during the pain state, and that were also altered in either microglia or in astrocytes. And I'll save you a lot of trouble. We came up with one single lipid target, and it's a 12-15 lipoxygenase product called hypoxylin B3. So if you look in spinal cord, this was significantly upregulated 2 hours after lipopolysaccharide treatment. It was also significantly upregulated in microglia over the different time points examined. And so we wanted to know what enzyme that this was being generated by. So it turns out that there are 6 enzymes in this family, and they're pictured here. And so we wanted to know which enzyme is actually making these 12-15 lipoxygenase metabolites, or 12-15 LMs, and then to investigate those downstream receptors. And there are 3 enzymes that are actually expressed in the spinal cord and in microglia. And I will save you a lot of trouble. It's 15-LOX1 that was changed significantly in microglia at the different time points examined. And so we wanted to know if selectively targeting this enzyme would then affect the pain-like behaviors. In collaboration with NCATS at the NIH, their chemical probes, ML351 and ML127, were examined in vivo. We administered these intrathecally prior to the delivery of intrathecal lipopolysaccharide. You should know that ML351 is much more selective for 15-LOX1, and it was also more potent, both in cells when we had tested that against heterologously expressed enzymes and also in vivo. So you see here, again, the area under the curve, significant allodynia with intrathecal LPS. And this was significantly reduced in a somewhat dose-dependent manner. I think we were probably at the top of the dose-response curve here. And this was also significantly blocked by the highest dose of ML127. And again, this was not affected at all by the non-steroidal anti-inflammatory drug, ketorolac. So clearly, this is something that is mediated by 15-LOX1, at least from the level of the drug. We also examined this in mice as well, because that was in a rat model. We wanted to know if this would also translate to the mouse, so that we could then, in the future, measure it in the rheumatoid arthritis model. So what we have here is in male C57 black mice, allodynia caused by intrathecal LPS that was significantly reduced by systemic administration of this inhibitor. And so this is an in vivo active inhibitor that is brain permeant. That was, shown before in the previous paper from NCATS, and we have demonstrated it now in the mouse model. In females, you can see it's completely reversed the established allodynia as well. So these are preliminary results. Obviously, these are ongoing, but this is encouraging to know that we can actually reverse the pain-like behavior of something that's involved in this chronification phase, since it's, rheumatoid arthritis is more prevalent in females. Okay, so, next we want to know, obviously, if 15-LOX1 drives the chronification in rheumatoid arthritis. So this is something that is ongoing. What I can tell you is preliminary data shows that measurement of the 12-15 LMs, 12 heat, hypoxylin B3, which I showed you earlier in microglia, and 15 heat are all increased during this chronification phase. So day 6 and day 15, this is happening at the right time. So future studies will examine this in the grant that was just received, if knocking out the gene or if the use of chemical probes will actually affect the rheumatoid arthritis state. So that's great. So, we know that targeting the enzyme could be a potential drug target, but we would also like to know what the downstream receptors are of these metabolites, because that could be potentially another avenue of intervention. So we asked, what are the receptors mediating the 12-15 LM induced pain state? So first we needed to know if these metabolites actually cause nociception directly in the absence of inflammation. So we delivered them spinally. This is hypoxylin A3 and 12-SHP, which have higher potency, so we measure those for signal transduction, but they're also a little bit unstable. And so in metabolomics, we've reported to you the hypoxylin B3 and 12 heat. And the short answer is yes. So this is measuring tactile allodynia. This is for all 4 metabolites. Compared with the saline vehicle, you see significant allodynia that persists for several minutes, even hours following a single injection. And these, I should mention, are concentrations that are physiologically relevant. So this is something that could happen in vivo. Next, we wanted to know, what the receptors are that mediate these effects. And so we examined the hypothesis that these potentially activate nociceptive receptors, TRPA1 and TRPV1. And this is based on the literature showing that these are activated by several different lipid metabolites. And so we asked our collaborators, Suzanne Doolin and Brad Taylor, who are now at Pitt, to do some calcium mobilization assays for us. And what you can see here is that after treatment with hypoxylin A3, this is sensory neurons, there is an increase in FURA2 fluorescence compared with the vehicle, and this is shown here. So we did this for TRPA1 and TRPV1 in both an overexpression, a heterologous overexpression system in HEC293 cells. These are untransfected and then transfected cells for both, and also in sensory neurons. And we showed this in rats, and this was sensitive to inhibition by antagonists for TRPA1 and for TRPV1. We also showed this in knockout mice. So we had knockout mice of either TRPA1 and TRPV1 and showed that that reduced the hypoxylin A3 induced calcium mobilization. So we were, and I should mention as well, we used the antagonists in vivo and rats and showed that those antagonists blocked the hypoxylin-induced allodynia at the spinal level. So this is, to us, provided very strong evidence that this is a relevant mechanism in vivo. And that's great, but it turns out that there are pretty significant side effects with systemic inhibition of TRPV1. Basically, the patients felt like their pain was relieved, but they developed significant hyperthermia. So, your pain goes away, but you feel like you're in the fiery pits of hell, right? This is not good, okay? And so we asked the question, and Christina Miliano, who is here, she was gracious enough to conduct the study for us. She measured core body temperature after systemic administration of either a TRPA1 antagonist, this is an Amgen compound, or ML351, which I showed you earlier, is an antagonist of 15-LOX1. And it turns out that antagonism of TRPV1 causes a significant increase in core body temperature, but there's no significant effect of blocking 15-LOX1. So that's encouraging. Obviously, we would need to see if there are any other side effects that are caused by this molecule, but this was encouraging to us. So the other question that we had is there potentially being other receptors that might mediate the effects of these metabolites. So if you look very closely at, these are the traces here for calcium mobilization, we apply hypoxylin A3, and there's a delay until we get an increase in FURA2 fluorescence, indicating increased calcium mobilization, right? And so we showed that, blocking TRPA1 and TRPV1 then reduces that influx. And just, for funsies, I'll tell you, we did potassium to make sure that the neurons were still alive, and CAPS is capsaicin to show you whether or not you have a TRPV1 positive neuron. In this case, it's a negative one. In this case, it's positive, so this one has TRPV1 receptors, which is important if you're using the antagonist. You want to know if it doesn't have an effect just because there's no TRPV1 there. But because of this delay, we thought maybe this is mediated by a GPCR. And those obviously act on a slower time scale than channels. And the reason why this was important to us is that 1/3 of all approved drugs from the FDA target GPCRs. So this is a very rich area for drug discovery. So what we wanted to do then is to be able to set up a screen for G protein coupled receptor activity. So Gilad Barnea years ago developed the Tango system. And basically, I'm not going to go into all the details, but specific changes were made to these receptor constructs so that once you have a ligand binding to a receptor, that then activates a tobacco H virus transcription factor to translate to the nucleus and activate luciferase expression in a specialized cell line known as HTLA. So these are basically modified HEC293 cells that have luciferase expressed once you have binding of your ligand. And so you might imagine that, if you have a ligand receptor interaction, then you would see increased luciferase activity compared with your vehicle. So Brian Roth at UNC has then expanded this platform to include all of the non-olfactory GPCRs. This is about 315 receptors. This is 88% coverage of the human GPCRome. And you might be asking, who is crazy enough to set up this assay? That was me. Without a robot, by the way. And the idea here is that, I'm not going to beat Brian Roth. I mean, he's got robots the size of the room, right? I don't have that. But what we do have is expertise with lipids. It turns out they're incredibly difficult to work with. They are hydrophobic, so they don't like to stay in solution. They don't follow Michaelis-Menten kinetics. They don't adhere to Lipinski's rule of 5, which makes them a little bit more difficult and challenging for drug development. And so, so I decided to do this. Thanks, Matt. Okay, so this is just an example of some of the data that we've got with screening hypoxylin A3. I showed you before. This causes nociception, increases calcium mobilization through TRPA1 and TRPV1 receptors. And now we want to know if it targets a GPCR. So this is a volcano plot. This is showing you the fold change versus vehicle, either positive or negative, and then the amount of significance. And we basically subjected this to anything that is above a 4-fold change from the baseline, because you can see you get a lot of hits, but they're not necessarily meaningful, right? So how do you filter that data? And that's one of the issues that you can have with omics data. What's interesting to us is that there are several targets that are actually involved in nociception, but one of them stood out to us in particular. It's this receptor known as MRG-PRD. So this is something that is exclusively expressed in nociceptive neurons. So that is pretty exciting for us, because obviously the ideal would be to come up with a therapeutic that targets the nociceptive response without affecting the reward system. So this is not expressed in the brain. Okay, and Ian Levine, who's probably in the audience somewhere, he's been helping me with these studies, which is fantastic. But basically, this is showing you a dose-response relationship, and as you can see, it's in the sub-nanomolar range, which is important because you want to know that this is a physiological interaction, right? There's a lot of evidence in the literature, for example, for this receptor in particular, that a ligand will activate in the micromolar range, which is ridiculous. I mean, that's not going to happen in vivo. So the published literature shows that beta-alanine activates MRG-PRD, but it's at 15 micromolar. So this is significant, and we feel like that's actually a real interaction. And we're pursuing that with follow-up studies to show the functional activation of G proteins, because as some of you might know, there are several different types of G proteins. So we're going to characterize this interaction further. Okay, so the important thing about this is that these metabolites activate 2 different populations of nociceptors. So the TRPV1 positive, which mediate heat-like pain sensitivity, and the MRG-PRD, which mediates mechanical pain sensitivity. And as I showed you, significant effects on tactile thresholds. So this is important to us. So the fact that we're seeing activation of multiple types of nociceptors means that this could be an important interaction in vivo. Okay, so in summary, what I told you is that rheumatoid arthritis is a chronic pain state mediated by spinal TLR4, and that early but not late phase inactivation of TLR4 reduces tactile allodynia. The acute to chronic pain transition is recapitulated by activation of spinal TLR4. This activation leads to an NSAID unresponsive pain state, so something that's not affected by non-steroidal anti-inflammatory drugs. So basically modeling a neuropathic-like behavior in vivo. It has a similar pharmacological profile to the chronification of RA pain. And that spinal microglia, at least in males, I should mention a lot of this data was in males, I showed you some data in females, happen in spinal microglia, but this might be a different cell type in females. So TLR4 activation in these cells leads to activation of 15-LOX1, which is a 12-15 lipoxygenase, producing these metabolites, which are pro-nociceptive metabolites. The inhibitors of 15-LOX1 activity both attenuate and reverse allodynia. Remains to be determined in both males and females, but those studies are ongoing. And that spinal administration of these metabolites directly produces allodynia in the absence of inflammation. And this happens via TRPA1 and TRPV1 receptors, and also via possibly a number of GPCRs with a predicted role in nociception, and this includes MRGPRD. I would like to thank the lab for the support, particularly Matt for giving me the space to be able to do this project, Christina for her, basically just helping out with everything. I don't even know where to start. She's a fantastic and very, very motivated and very bright postdoc, and we're very lucky to have her. Laura, who's a graduate student with us, who has just done a fantastic job with behavioral assays. She has so much hustle. Ian Levine, who's here in the audience, just, stand up. He's just done an absolutely fantastic job. He's basically running cell culture assays by himself. It's just incredible. And that's a tribute to the program that we have here. My collaborators, my previous mentor, Tony Yaksh, my postdoc advisor at UCSD, and Ed Dennis, our collaborator. Camilla Svensson, who's now at the Karolinska, who's just an incredible leader in the pain field now. Our collaborators at Pitt, Suzanne and Brad. The team at NCATS for providing the compounds and the support to be able to follow up these studies. It's exciting to be able to be involved in drug discovery at this phase. I'd like to acknowledge our funding. I should mention Ian has his own funding. Good for you. And if we have a couple of minutes, I don't know, I could kind of go into a little bit about the future directions. I mentioned some of them already. One of this is extending the behavioral measurements of pain, because historically in the field, a lot of them have been evoked measures, where you present a von Frey filament to the paw, or, thermal escape. So you measure sort of like an escape response. And while those measurements are very robust, we don't typically do that with humans. We don't follow people around with hot plates and stick them with von Frey. You might stick them with von Frey hairs, but the point is that, a lot of this is just spontaneous. You feel terrible. You're curled up on the couch. You don't want to talk to anybody. You may have changes in functional measures. Like with arthritis, for example, people lose grip strength. They can't open doors. They can't pick things up. They have trouble working. And so our animal models should really reflect that. And that's one of the reasons why I came back to academics to refine those sorts of measurements. And so we're going to be doing that with the arthritis models. We're going to be using, as I mentioned, males and females. And also to determine the cellular sources of this 15-LOX enzyme, and if there are contributions of the other 12 lipoxygenases as well, because that's possible. And then also, as I mentioned, looking at the downstream GPCR targets of 12-15 LMs, and this is the work that's being supported by a recently awarded R1 and the CHRB grant from the state of Virginia. So you can imagine that this could then be extended to other pain models. And in fact, this is something that you do in drug development, is to have multiple models supporting your data, particularly with neuropathic pain, because it is so complex and such a diverse set of mechanisms that can happen. This is an example of chemotherapy-induced peripheral neuropathy. So it turns out that about half of patients who receive chemotherapy develop pain, and maybe half of those will actually consider quitting the treatment because the pain is so severe. So this is an example of a chemotherapeutic, paclitaxel. If you give it systemically to animals in the same formulation that you give it to humans, it causes significant allodynia. This is something I did in industry, so I can't tell you what the target is, but if we give an antibody targeting this lipid, it completely reverses the pain state. And this is blinded, as it should be. So this was encouraging data for us. And if you switch it to the control antibody, that therapeutic effect goes away. And then if you then just administer the control antibody, the animals are still allodinic, and then you can reverse that by doing the crossover experiment. So this is to show that, targeting lipids could be a good therapeutic avenue to explore. You can also look at measures of peripheral nerve injury. So this is spinal nerve ligation model. You could also consider incorporating higher structures in pain. Obviously, I've told you a lot about the spinal cord, about sensory neurons, but it turns out that the brain is also very important. So we could extend our metabolomics methods to measure other regions. We can do microdialysis in the laboratory. A lot of the things that I've told you about are basically in tissue. If you do microdialysis, you can implant a probe in a specific region of interest, such as a region that's mediating pain states. You can measure signaling competent molecules that are released into the extracellular space that could potentially activate your G protein coupled receptors. We could also extend these methods to include other eicosanoids and perhaps endocannabinoids, other lipid classes, and neuropeptides. So you could sort of see this turning into like a discovery-based platform where if you have a ligand without a known receptor, you can then screen it in your against your GPCR library, and then translate that back in vivo, develop antagonists to this, screen it in the same GPCR screen, and then see if your drug will affect the behavior in the animal. And before I go, I just wanted to leave you because I think this is so cool. And Christine Founts is actually working on click chemistry in the lab as well. What we can do in collaboration with others, so this is from with Megan Matthews at UPenn, you can actually turn your drugs into probes and visualize where they bind in the cell. So you can use this process called click chemistry. It's basically a process that's like, it's running orthogonal to other biological processes, so it doesn't affect normal biological processes to turn this probe into an alkyne, and then you can expose it to your cells and react it with a rhodamine-based tag that is an azide, and through that reaction, you can then see where the drug is binding in the cells. So that's shown in red here. So we're pretty excited about the possibilities of doing this in the future. So with that, I'd like to close, and I'd like to thank you all for being here. Thank the School of Neuroscience for inviting me, and I'll take any questions that you have at this time. Yeah. Thank you.
So the GPCR you found,
Yep.
is expressed in hexels because you just put the channel in hexels and you saw.
That's a great point. So actually, the screen that I mentioned, those receptors are codon optimized so that they are expressed differently than it will distinguish from whatever's endogenously expressed in the cells. It turns out in hex cells, the expression of this receptor, it's very low or non-existent. But that's how you would distinguish between the two when you overexpress it. Oh, you transfect them.
Yeah, but
Yeah.
so you need to act directly on the channel.
Yes. Well, okay. We think that there's possibly a direct interaction, but you would need to do electrophysiology to really answer that question. And we think because we also have activity of a GPCR that it could be that it activates the GPCR first, and then there's a downstream interaction with the TRP receptors. But yeah, we would need to do more studies to see the nature of that interaction, but that's a great question. Yeah.
What do you think the GPCR downstream signaling pathway to activate TRP channels?
So as far as the G protein, so that one in particular, MRGPRD, is known to couple with GIGO. That's the same as opioid receptors, actually. So it's, I mean, without doing those studies, it's unclear at this point. But there can be activation of, MAP kinases through small G proteins as well through that pathway. So, that's a possibility that could be explored. We don't know yet, but that's what the future studies will be designed to do.
Question.
Yeah.
You already touched on this, but maybe just can elaborate a little bit. So you're talking about how obviously pain is process at different levels, spinal cord, brainstem, up through the cortex.
Yep.
And you're focusing a lot on the spinal cord. Maybe just even from the industry perspective, is there advantages to focusing there? Like, is it the first step and it just gets too complicated when it gets to the brain?
Yeah.
Or no, or
Yeah.
like the reward system. I mean,
No, I mean, that's a great question. I mean, honestly, like where I was as a postdoc, you know, the main focus was on the spinal cord. And I think because it's the first central synapse, right? So this is like the first place where you see central sensitization take place. But obviously the brain is critical, right? And so that's why I mentioned the future studies would include looking at different brain regions to see if this is really a relevant mechanism in vivo.
could that be part of the chronification? Like if once you
Exactly.
have injury or whatever
Yes.
caused the pain to begin with, is there chronification because you're having changes from the brain that are coming down and then like modifying those circuits
Yeah.
or something?
I mean, this is the age-old question. I mean, this is, you know, you've hit on the major question in the pain field is like, yes, that is important and we need to do it clearly, you know, because we don't know that just affecting something at the spinal cord is going to, quote, fix everything, you know. A lot of approaches right now in treating pain are sort of multimodal. You know, you'll combine different treatments together, and that's probably going to be the most likely scenario moving forward. I mean, do I think like this is, you know, the magic bullet? No. I mean, yeah, it's an important step, but, you know, it may be sort of like a combined therapy type of thing, or, you know, it may be the fact that, you know, that it's exclusively expressed in nociceptors. We can just bypass the reward system entirely, which would be fantastic because, you know, as you know, it's sort of the holy grail to develop an analgesic that doesn't cause addiction, right? So.
Any other questions?
Very nice talk.
Thank you.
You know, with the GPCR stream, you know, got this great candidate to look at. There are a few others though. In there.
Yeah.
The possibility of redundancy built into the system. I mean,
Yeah, that's a great question. So you know, the idea that these are metabolites that affect multiple populations of nociceptors is intriguing. And in which case, like, yeah, it may be more effective to block the enzyme, the production of the metabolites to begin with, right? Is that going to be NSAIDs 2.0? I don't know, right? But this is why we're looking at it from multiple angles. Like if you block the formation of the metabolites by inhibiting 15-LOX1, that may be a more effective approach because if you just cut off the production of all of these metabolites, you know, it could then, you know, potentially block all of the downstream interactions, and then you don't have to worry about giving multiple drugs. So the chemotherapy data that I showed you, that was done in industry. So I can't tell you what the target is because then I'd have to kill you. But I don't know. The company's not around anymore, so maybe not. But what they were actually doing was they were developing monoclonal antibodies against lipids, and so this way it's sort of like a sponge or a sink, you know, to then block the downstream interactions with all the different receptors. So whether or not that's yeah, I mean, so that would you would sort of achieve a similar thing by blocking the enzyme, the production of them to begin with. So yeah, that's a fantastic question.
Okay, I guess that's it.
Thanks, guys. This is great.
I feel the same way. You never do it.
Oh, thank you. Sounds good. Thanks so much. Thanks for coming. Hello. Hi.
I apologize for that.
No, you're fine.
She's got me all shaken up.
Sorry. Don't be sorry. Come on. That was great. You got all the things right, you know? Like,
I gotta go eat lunch.
okay.
Do you want to hand me my pointer so I don't mix them
will
up?
hand
Is this
you-
mine?
That's yours,
All right.
because I used yours. I have my last Oh, yeah. I don't know.
They usually clear out.
Oh my god, pacer chat, yeah. When I moved up here, I was like, what is this test? We didn't have no pacer test.
side?
This side over here?
Yeah, it was a,
I see something over here. It's not a beanie.
no, that's not a beanie. No, I don't see no beanie. Okay, I said I would look, and that's what I've done.
Yeah, it's
No
hard
one's
to tell.
found it